Okay, so my name is Tianna, and I am a second year student at the University of Winnipeg. I've had the pleasure of working under Dr. Doucette for now the second summer in a row, and the project I've been working on is titled The Effects of the Hepatic Nuclear Factor 1 Alpha, or HNF1 Alpha, G319S polymorphism on insulin secretion. So to start off, I'll just give a little background information on youth onset diabetes in Canada and then the HNF1 alpha polymorphism itself. So diabetes in Canada is increasing at a very alarming rate, with the majority of cases being type 2 diabetes. Type 2 diabetes is characterized by the development of insulin resistance and failure of beta cells to secrete proper amounts of insulin. It's often associated with obesity, a sedentary lifestyle, genetics, and it's typically been seen, or traditionally been seen, as a disease affecting only adults. However, we're seeing it now more and more increasingly in children, especially here in Manitoba. Actually, Manitoba has the highest rates of type 2 diabetes in children in Canada, which are over 22 times greater than the national average. In fact, the very first case of type 2 diabetes diagnosed in a child in Canada was diagnosed right here by Heather Dean in 1985, which really wasn't that long ago. Here's some results from an older national surveillance study looking at the incidence of type 2 diabetes in children for 100,000 children in Canada. And so you see the national average is just about 1 or so, but then you see in Manitoba it's greater than 12. And then here are results from a more recent localized study. So again, here's the national average, still around that, just above 1 between 2006 to 2008. And then from 2006 to 2011, you see the increasing rate of type 2 diabetes in Manitoba. And I'll also add that the majority of children affected by type 2 diabetes in Canada are of Indigenous OG descent. So with such a large specific population affected by this disease, that gives us indication that there's probably some genetic element to what's going on here, which leads us to the polymorphism of interest to us. So wild-type HNF1 alpha is a gene that looks for transcription factor that targets many genes involved in glucose metabolism, such as those listed here, which are involved with glycolysis, glucose transportation, insulin exocytosis, and beta cell growth. And HNF1 alpha was first discovered in the liver, hence the name, and then later in the pancreas and other tissues. The polymorphism variant, which I should add, a word polymorphism is a mutation of a population of people. So in this gene, there's a single nucleotide substitution that leads to a substitution of then a glycine with a serine at codon 319. And it just so happens that glycine has a neutral charge and the serine has a positive charge. So this has implications for the folding and structure of the protein, and then in turn the function possibly. It was first identified in Indigenous OG populations in northwestern Ontario, and then later in the similar population here in Manitoba, so it's specific to that population. Among Indigenous youth that have type 2 diabetes in Manitoba, about 40% of them have at least one copy of the G319S variant, and it seems to have a dosage-dependent effect. They saw in the adults in northwestern Ontario affected that about, if they were homozygous for this variant, that you'd be seeing development of type 2 diabetes in about the 3rd decade of life. If they're heterozygous, in the 4th decade of life, and then it's those without the polymorphism, you normally see development of type 2 diabetes in the 5th decade of life. For those here in Manitoba, those affected by the polymorphism, they appear less obese, they have less insulin resistance, and have less plasma insulin than those without the polymorphism, which gives us some indication to how accelerated the disease is in these people. They're no longer needing to reach such older ages and high levels of insulin resistance before developing the disease. It also gives us some clue that there must be something else going on here. For example, this polymorphism and beta cell failure. Despite the association being made between this polymorphism and the development of type 2 diabetes, its impact on function of the pancreas has never before been studied at a cellular level. So that leads me to what I began working on last summer. The project, or question we were looking at was, how does the HNF1 alpha G319S polymorphism impact insulin secretion in the pancreatic beta cell? And how we did this is we used a clonal mouse cell line with insulin secreting properties, and then we transfected them with empty vectors and negative control, wild type HNF1 alpha, or positive control, and then the G319S polymorphism. We then went on to perform glucose-stimulated insulin secretion assays, or GSIS assays, where we took the cells, we put low glucose on them, incubated for about half an hour, let them secrete insulin. We then collected that to be analyzed later, and then did the same thing with high glucose. Put that on, let them secrete insulin, and then collect that. And then we also collected for DNA, just as a normalizing factor in our calculation. And then we analyzed those low glucose, high glucose samples for insulin secretion via ELISA. And here what we found, so we have the empty vector here, the wild type HNF1 alpha, and then the G319S. And we found that we have significantly higher basal level secretion in the polymorphism, and then also a trend towards lower glucose stimulated secretion in the polymorphism. What's most interesting is when you look at the difference between the low glucose and high glucose secretion in each of the areas here, which I just have on the next slide. This is the one I had just showed you previously. So this shows the fold change in secretion, and here now we are getting a significant decrease in secretion in the polymorphism. So, in quick summary, we did find noticeable changes in insulin secretion in the G319S polymorphism, and it's really that decreased stimulated secretion that we're interested in. So, big question, why is this happening? And that's what we're trying to get into this summer. So what's causing this impaired glucose-stimulated insulin secretion in the polymorphism? Before I get on to going about how we did that this summer, or the question we first tried to tackle, I'll quickly go over different insulin secretions. So basal secretion is that constant low-level insulin secretion between meals while you're sleeping. And then glucose-stimulated is basically that insulin secretion response above and over that basal level after you eat a meal, for example. So that's why we were doing those low glucose and high glucose treatments, just to see, to observe basal secretion and then stimulated secretion. And then I'll also go through how glucose-stimulated secretion works. So glucose, you eat a meal, glucose enters the beta cell. It's then broken down by glycolysis to pyruvate, which is then further metabolized in the TCA cycle, electron transport chain, which creates a proton motive force, which provides energy for ATP synthesis. ATP then inhibits the K-ATP channel, which depolarizes the cell. And then you have an influx of calcium, which signals for insulin secretion. And by going about this route of aerobic metabolism, you generally produce around 36 ATP molecules here. And then, so this is what we were working on for the majority of my summer. Does lactate dehydrogenase, or LDH, play an important role in altered insulin secretion in beta cells that express HNF1 alpha G319S? So a little about why we chose LDH, or what it does with glucose metabolism. LDH is ubiquitously expressed, and it's a shunt away from that aerobic metabolism. When LDH is activated, it reduces pyruvate to lactate, so it no longer continues down that long cycle to produce 36 ATP molecules and insulin secretion. It shunts it away, and you have lactate, and then it generates a net of 2 ATP molecules. So it's just quite significantly reduced. So you can imagine that would have a great effect on insulin secretion. And just, again, the figure I showed you before, instead of following down here, it's shunted this way to lactate. So this was the first evidence to suggest that LDH might be doing something. That's why we kind of took it and ran with it. So we did a Western blot on a few different proteins involved in glucose metabolism, and LDH was the only one that we saw increased in our G319S polymorphism here. So just by visualizing here, you see the upregulation of LDH in the polymorphism. And this is just a housekeeping gene for controlling of the equal loading of the gel, and then this is H-type-1 protein, which just confirms that our transfections were successful in both of our wild-type polymorphism there. So then we took this and we wanted to quantify it, so we used densitometry, and we got here that there are significantly higher levels of LDH in our G319S polymorphism. So now we want to answer the question, does LDH have an effect? And we did that using two approaches. The first is by an LDH inhibitor. So if LDH is playing this key role, if we inhibit it, we should be able to restore normal glucose-stimulated insulin secretion in the polymorphism. So we did the same approach, took the same cells, transfected them. Only difference is we treated them with oxamine, which is our inhibitor before performing the GSIS assay, and then collected and again measured insulin secretion. I unfortunately don't have any results to show you from this yet, as we're not as far along as we'd like to be, and I spent the majority of the summer kind of working out protocol, concentrations, timing, but hopefully soon. And while we were in the midst of figuring all the protocol for that out, we also started another approach, which was focusing on the ATP. So we decided, again, the same protocol here, the only difference is afterwards collecting for ATP instead of insulin, and then measuring ATP. So we hypothesized that the G319S should be producing less ATP, as if LDH is playing a role and shunting it away that way to produce lactate, which could explain our decreased insulin secretion. I unfortunately don't have any results to show you for this either, because I'm actually just in the middle of the assay for my first samples. But again, we're really excited for those results, and hopefully it all turns out. And then just some future directions for the project. First would be to produce stable cell lines using a new gene editing technology, CRISPR-Cas9. By doing this, I wouldn't have to transfect cells for every experiment. It reduces variability and it's time consuming. So by that, I could take that all away and just have cells ready to go anytime we're ready to do an experiment. And also, the transfection method isn't the best because it's overexpression, and it's overexpressed in proteins in levels they wouldn't normally be. So by having just this stable cell line, we can have protein present in normal amounts and also still have our polymorphism there too. Also, we could identify changes in the HNF1-alpha G319S targets by qPCR. For example, those targets I mentioned earlier that were involved with glycolysis, glucose transportation, and all of that. See if something down the line there is affecting insulin secretion. Also, understanding those higher basal levels, because we're not quite sure why the G319S secreted significantly higher than the others. And also not a lot is known about basal secretion in general, so we're kind of interested to find out why. We can also look at different nutritional influences, such as what a high-fat diet does to the cell, or starvation. Actually, last summer started looking at the high-fat diet, and we did see significant results, so just continuing on that and see what happens is in the plans. And then, of course, a mouse model, which is on the way, hopefully soon. It will be able to give some more physiological relevant data. We'll be able to manipulate diet and pregnancy and look at a whole bunch more items there. And also, cells aren't always that reliable. So hopefully in the coming months we'll have that soon. And then I'd just like to thank my supervisor, Dr. Doucette, for all her guidance and help over the summers, and the rest of my lab for listening to my presentation, answering all my questions, and then Dr. Barb to explain for the plasmids used in our transfection, and then, of course, for all the funding over my last 2 summers here.
Questions?
Yeah, really excellent talk. It's really nice. So just remind me, what does HNF1-alpha actually do? What's its function?
It codes for a transcription factor, which targets a bunch of genes mainly involved with glucose metabolism. So that's where, not sure how to do that. The ones I have listed here, which, again, I'm not sure which ones are which, but some are involved with glycolysis, glucose transportation, insulin exocytosis, and beta cell growth.
Right, so it binds to DNA, right? So how does it bind to DNA? In what kind of form? Is it just like a single protein that binds to DNA, or is it more?
No, there's two. I'm not too sure, to be honest, but I think Christine.
So when you're doing your transfections,
Yeah.
like you're using this cell line, this Min6, right? Yeah. So is there HNF1 alpha in those Min6 cells already?
No, we're transfecting them in.
Right, but just
Yeah.
even without the, like you have your
Oh,
empty
yeah,
vectors
originally?
control, is there HNF1 alpha in those cells already?
That's a good question that I should know. I believe there is.
Okay, yeah, I think there probably is. So I guess my kind of question is, like, when you're transfecting it in, what do you think the impact is? Like, how do you account for what is binding to the DNA? Is it, if there's two binding, are you getting like a wild type HNF1 alpha and a polymorphism, and the DNA is a heterodimer, or is there just two polymorphism type or variant type of HNF1 alphas binding, or do you have just wild type? You know, do you have those three combinations, or do you just have one?
I'm not quite sure what you're referring to, but I know they should be separate because we have the individual plasmids for each, and then we transfect them separately.
Right, but if in your EV group you have HNF1 alpha
already in it.
when you transfect the G399S,
Oh, there would be both. I see what you
you
mean.
probably have both, right?
I see what you mean. Yeah.
So how would you account for that? Do you have any idea? Understanding what's binding?
Yeah, that's a good question. I'm not too sure, and that's not something we've done yet. But we probably should be looking into how to do that in the future.
Yeah.
As to ways to do that, I'm not exactly sure.
Okay, yeah, fair enough.
Yes.
That's really good. Do you know of any inhibitors that might repress HNF1 alpha transcriptional activity?
I am not aware of any first with it, I'm sure.
Does HNF1 alpha, does it activate the transcription of lactate dehydrogenase?
No, it doesn't.
I guess maybe he was going forward, and this is a question that I just thought about was, have you tried doing knockout studies?
Pardon me?
Have you tried doing knockout studies, like gene knockouts?
Oh, knockout studies? No. Not that I'm aware of.
Any further questions? Not? Did you have anything?